That's what I started doing now. So we will continue where we stopped last time, and now this is like a real physiology where we talk about proton multi-force generation and the 4 ways to generate proton multi-force. And all these things are quite important to understand everything else, especially bioenergy. So, because all unit is called the membrane system of microbial cell, of membrane systems of microbes, we are talking about a membrane as a place where this proton motive force is happening at its event. So, bacteria mostly use membranes, well, proteins that are inside of the membrane to generate PMF or proton motive force. So PMF or proton motive force has two components, electrical and chemical, and we will cover both of them, but generally it is based on chemiosmotic theory. What means chemiosmotic theory? So in chemiosmotic theory, that means the cells are coupling reactions that are generating certain energy with reactions that will utilize that energy. So basically, energy generation coupled to energy utilization. So that is chemiosmotic theory. So proton motive force or PMF is creating a H plus gradient or proton gradient, and that gradient is created across the membrane. So there is a difference in concentration of positive ions in and inside of membrane or H plus ions, and that gradient is later used to synthesize ATP, and ATP is then used in later biosynthetic processes downstream. So those other processes downstream of ATP production are membrane transport, multi-drug efflux pumps, flagellum rotation, or anything that needs ATP molecules. And these multi-drug efflux pumps, solid transports, we will cover in the lecture that was scheduled to be lectural today, but because we didn't finish last class, we are going to talk about that now. So, as I said, proton motive force has two components, electrical and chemical. So what means electrical component proton motive force? So that means that H plus pumping, like protons, create positive charges outside of the cell and negative charges inside of the cell. So basically, positive out, negative in. So that's the gradient between that refers to the electrical component. The only exception to this rule are organisms that are called acidophiles, and we will talk more about them when we talk in Unit 5 about microbial stress response. So in case of acidophiles, it's opposite. Positive charge is accumulated inside of the cell. So they have an invert membrane potential. Again, in Unit 5, it will be more clear. So this part of proton motive force is called electrical component, and electrical component refers to the charge difference between outside and inside of us. So outside will be positive, inside will be negative. That proton gradient, positive negative gradient will be used to create ATP. In terms of electrical component, we have a new term called membrane potential, and this is the delta stands for the change, psi, delta psi. So this difference in charges, positive out and negative in, it's called membrane potential, and we express that in millivolts. Again, negative inside, positive outside, and except in case of acidophile. In case of acidophile, situation is slightly different. So, membrane potential is stronger if the difference between charges inside and outside is stronger. So that's where that difference will determine membrane potential to produce certain energy. How you measure membrane potential? Again, we are still in electrical component of proton force. There is a very simple experiment to measure electrical components of proton motive force or membrane potential. So you will take this positive charge, so simply positive components that will be stuck to the membrane outside. And then you will, in order to do that, you will have these lipophilic cations because they will create that positive charge. Why they have to be lipophilic? Because they have to pass through the membrane. And then because they will be positive, they will be drawn from the outside to the inside, where the environment is negative. So that's why they are positive lipophils. So they will just go down through the membrane because membrane is hydrophobic. And what you are measuring with this probe, you are measuring the speed of these, in which these cations are transferring from the outside to the inside of the membrane. So when equilibrium is reached, that means that we are all, literally all positive came inside and now it's equilibrium. So when equilibrium is reached, the charge will exist outside and inside the membrane, and that difference, there will be this difference that you will measure as a membrane potential. So that's how we measure electrical component. And again, please don't memorize this formula. This is just the way how it's calculated if you are measuring. Very similar to the electrical component, but observed from different perspectives, is chemical component on proton force. So the thing how electrical component relies on positive, negative differences from outside and inside the membrane. Chemical component on proton force is relying on concentration of H plus ions between different cellular compartments. And H plus ions are referring to the acidic compound, while negative ions, OH ions, are referring to basic compound. So the chemical gradient that exists between two compartments allows ATP synthesis at the end of electron transport trains operate. Again, that will be way more clear when we talk about electron transport train. And what you measure delta pH. So in the electrical component, you had the delta psi, which was membrane potential. Here you measure delta pH, which is the difference between concentration of H plus or acidic ions intracellularly and extracellularly. That's why we call it chemical. Similar to the electrical component, if you want to measure the chemical component of the proton motive force, so you will use acid or base analog. So we don't have any more lipophilic cations. This is analog cations. So you will use weak acid or base, and acid or base will freely diffuse across the membrane. And once when acid and base is inside of the cell, it will dissociate, and after it dissociates, it will create H plus and OH ion inside of the cell. And based on that H plus OH ions, cellular pH will get changed or altered. And depending on the cell pH, depending on the pH inside of the cell, if pH inside of the cell is more alkaline, that means it has more OH ions than inside, then you will, so if that's the case, if it's more alkaline, then you will use weak acid that has H plus ion, because you want to make sure that ions are diffusing across the membrane. If pH inside of the cell is more acidic, that means it has more H plus ion than outside, then you will use weak base to measure chemical component of proton motive force. So in past, people used radioactively labeled acids and bases. Nowadays, they just conjugate fluorophore to acids and bases, and then they can track it in real time. What's happening and what's the chemical component of proton motive force. So this is very easy. In summary, what we learn about proton motive force is that to measure the difference between pH, to measure the difference in pH between outside and inside of the cell, there must be differential diffusion of weak acid or base across the membrane, usually weak acid. So if delta pH is 0, the difference, that means that difference in H plus ions doesn't exist. So can you still measure chemical component of proton motive force? The cell can cell actually still can be active and generate PMF. How? So if there is no difference, well, even if there is no difference, cell still has membrane potential because membrane potential is not only based on diffusion of H plus ions, it only relies on charge the moment. So if you think of what else can be monoplus charge, you will see that there is this sodium gradient that is used to give delta psi, which is a membrane potential. So cell can still create proton motive force, but in this case, proton motive force will not be based on H plus ions, it will be based on sodium ions. Usually alkaline cyclic organisms use this. We will talk about that in a minute when we talk about cellular stress. So how is proton motive force generated in cell membrane? Again, everything is happening in cell membrane, and we will go step by step to make sure that everyone is on the same page how proton motive force is generated. So this is just a brief, simplified picture of how electron transport chain look in the membrane. So it's nothing else but a set of proteins in a membrane that are accepting and releasing electrons, and that's which is followed by proton diffusion. So proteins that are running and that are involved in electron transport chain, or ETC, have ability to extract and pump protons from cytoplasm to periplasm. So that's called a vertical transport. So what precedes pumping of protons is translocating of electrons in the membrane through various carriers. And you will see that very often, oxidation and reduction. Oxidation means releasing of electrons. Then when one carrier releases electrons, then the other one is accepting it and gets reduced. And that electron jumping is followed by proton translocation across the membrane. And then that's how you get that gradient. So the high at the beginning of electron transport chain is a high energy electron. And in aerobic organisms, that is usually an ETH. But there are also light involved in the reaction that are boosting the electron into the higher energy states with the electron transport chain can start. And you will see through the electron transport chain proceeding that substrates are constantly oxidized and reduced. So whenever something releases electrons and gets oxidized, something else in the membrane will accept those electrons and become reduced. And then now this fully reduced compound will release those electrons and become oxidized and give that electron to another compound that becomes reduced. So this electron passage from carrier to carrier is happening in the membrane. And during that transfer of electron, the energy, certain energy is lost during the transfer. And that loss of energy is used to drive protons outside of the cell. So that's how you get creation of that PMF, or proton motive force. That's what we talked about a few minutes ago. So that proton gradient, which is proton motive force, driven by energy loss from electron transport, is creating a gradient, and that energy, the gradient that is contained in that gives that energy, is used to synthesize ATP from ADP and PI in the end of electron transport chain. So very often you will hear that ATP synthase in the end of transport chain is called complex 5, and that's where a big portion of energy is created. So this is where we would end the lecture last time, but I'm going to now stop this lecture and share another one. Any questions? I think I stopped my screen sharing. Any questions? And probably many questions. So this proton motive force that we just talked about something that will be very important when we start especially when we start talking about cellular pathways, which are in unit 6. And I don't know how to do this. Oh, I got it. No worries, I got it. Haha. Got it. I'm so proud of myself. Finally, I didn't mess up. Yes, see, next year I will actually know how to use technology. In 10 years, I might even learn how to do more complicated operation. So now when we understand proton multiforce, we understand chemical and electrical components. So this will be very important for unit 5 when we talk about acidophiles and alkaliphiles, because some of them rely only on delta psi. Some of them rely on both delta psi and delta pH, and it's very simple. They're trying to defend themselves from environmental stress. So how is this proton multiforce gas? So I briefly told you what is electron transport chain. Transport of electrons from carrier to carrier inside the membrane, which is followed by proton translocation across the membrane, and the gradient contained, the energy contained in that gradient to be used for ATP synthase at the end of it. But unfortunately, not all microbes are able to generate proton multiforce in this so-called simple way. So that's why we are covering 4 different, well, actually 5 different, but we are mostly we them in 4 mechanisms of generation of PMF. So one is a redox loop mechanism, which is this simple electron transport chain. And I say simple, even though it's not very simple, because that's the most common and that's what we all learned and we all understand. So all those proteins inside of the membrane complexes 1, 2, 3 are called redox loop mechanisms. It's all. Then you have something called direct hydrogen or H+ translocation. This is common for certain organisms, like light-dependent organisms. It involves light regulation, and cytoplasmic proton utilization will be something that you will see in organisms that are trying to defend themselves, that are able to survive in highly acidic conditions because they just don't want to open their gates. Because they prevent themselves from a lot of hydrogen ions coming in, so they deal with that in cytoplasm. And you have something called periplasmic oxidation of substrate. Separate way of doing it. Sorry, I have to correct myself. Direct hydrogen translocation is not related to light-dependent organisms, but I will. Let's go into it. It is, but it's not only. It's a fifth. There might be other ways to generate proton multiforce. But let's stay first with a redox loop mechanism. So what is electron transport chain? So there are certain complexes, complex 1, 2, 3, that are redox loops. So how all process goes? So electrons from some organic compound will arrive to the complex 1. Complex 1 is called NADH dehydrogenase. Whenever you see the name dehydrogenase, you will know that this refers to oxidoreduction reaction of something. So something is going to be oxidized, aka release electrons, while the other something else will take those electrons and become reduced. And that's usually done by enzymes dehydrogenases or oxidoreductases, even though they're not always quite same enzymes. So NADH dehydrogenase is a complex 1. So the one that is oxidized in this case is NADH. NADH is highly energetic. It's reduction equivalent. And NADH dehydrogenase is the enzyme that extracts electrons from NADH. And all this is happening actually now in cytoplasm at the beginning of electron transport phase. When NADH is oxidized and releases electrons, it becomes NAD+. So that's the oxidized version of NAD. So now this electron needs to be given to something else. What is happening at the same time with electron extraction is a translocation of proton ions from the cytoplasm. Because NADH dehydrogenase is also proton pump, unlike the other enzyme in the line. So this is first complex. So where these electrons that are extracted from NADH, where do they go? So they go to the second complex in line, which is called complex 2, simple as it is. The complex 2 is linked to the ubiquinone, and ubiquinone is very important electron carrier that floats in the membrane. It's tightly linked to the complex 2. So when ubiquinone accepts electrons, it will become ubiquinol. It becomes reduced. And those electrons will come from complex 1. Now ubiquinol is full of electrons and it's ready to move on. Another part of this complex 2, which is actually tightly linked to complex 2, but it's in cytoplasm, it's part of TCA cycle. Again, TCA cycle will be mentioned in Unit 6. So the enzyme from TCA cycle called fumarate dehydrogenase is oxidizing, succinate dehydrogenase is, as the enzyme, as the name says, it's oxidizing and reducing something. So in the process of succinate dehydrogenase, FADH is oxidized, releases electrons, and it gets to be FAD plus, and those electrons will be delivered to quinol, to complex 2. And the way how I used to imagine cells when I was young undergrad student was, this is a cell and there is a TCA cycle in the center of cell. But that's actually not how cell looks. So if you ever saw TCA cycle, it's like a little circle with a bunch of enzymes. So the way how cell actually looks is those circles are stuck to the membrane because this succinate dehydrogenase is going to do oxidoreduction and electrons extract have to be delivered to quinol. So it's not like a one TCA cycle. There are many, many, many circles that are stuck to the membrane. So that's like a big bubble burst for me when I realized that it's actually not the way how I imagined it, quite different. And that was also nice. That Lisa, actually, Dr. Stein taught me that. So again, complex 2, unlike complex 1, is not proton pump. So there is no generation of, there is no protons that are transported over membrane or complex 2. So now from complex 2, from ubiquinol, now it's reduced, it has electrons. Electrons need to move on to complex 3, which is, in complex 3 is called cytochrome oxidoreductase. So the name tells you that there is oxidoreduction involved. So this is that redox loop process. So in complex 3, which is oxidoreductase, so as the name says, cytochrome oxidoreductase. So the substrate for this enzyme is cytochrome. Cytochrome is membrane protein. It's not integral membrane protein, but it lives in membrane. So it's not integral like receptors, but it is a protein in the membrane. So this cytochrome oxidoreductase acts on this cytochrome in the membrane, and cytochrome is very specific protein that contains irons. So iron in those cytochromes will accept electrons, and iron in cytochromes is initially 3+, but when it takes electrons and becomes reduced, it's getting to be 2+. Now it's reduced. So and this is very rapid, when electrons now get released from this iron in cytochromes, so now iron again becomes 3+, and this is oxidoreduction. So everything is in cytochrome is done by cytochrome oxidoreductase, and it's happening on iron. So it's like a oxidoreduction from 3+ to 2+, and then from 2+ again to 3+. Those electrons have to go somewhere else. Complex 3 is same like complex 1, also pump protons across the membrane. And this is the end of redox loop mechanisms. Next way of proton multiforce is also tightly linked to electron transport chain, but we call this direct hydrogen translocation. So and there are two ways of this. That's where I kind of got a little lost. So one way of direct translocation is related to complex 4 of electron transport chain. So after cycloperoxidorelapse. So this is terminal oxidase in electron transport chain. But second way of direct hydrogen translocation as a mode of proton-multiforce formation is bacteriorhodopsin. This is light dependent. So it's also direct hydrogen translocation, but rhodopsin and bacteria that do that, which is rhodobacter, have their own way of proton multiforce generation. It's called direct translocation. So what is terminal oxidase? So if you remember from complex 3, now that's cytochrome oxidoreductase, so cytochrome now released electrons, iron became 3+. So now electrons go to the complex 4, and that's cytochrome oxidase. So what this complex does, it literally, this is almost the second last in electron transport chain. So this complex actually holds the oxygen. If we talk about aerobic respiration, and we do in this case. So the complex holds oxygen tightly. And what all enzyme has, it has iron and copper. So think about iron and copper as the two hands that hold oxygen. And that oxygen now accepts electrons from complex 3. And it holds oxygen until it's completely reduced to form water. And it also pumps proton out. So the way how you pour water is like this oxygen picks up two hydrogen ions from surrounding media inside the cell, and you get water to be made. And this is also proton pump. So you have proton pumping on complexes 1, 3, and 4. Complex 2 does not pump protons, but these 3 complexes in aerobic organisms are enough to create proton gradients that can be used as energy to create ATP. When we have, we will see when we talk about alternative mode of respiration in E. coli, there is a change in terminal oxidase. It's called reductase, but let's not get confused yet. There will be enough time. Another way, a specific way of direct hydrogen translocation is light-activated bacteriorhodopsin. And for that, you have light that, like photon of light, that like absorption of that photon that releases energy. So now the energy doesn't come from oxidation of organic substance, it comes from light. So now this absorption of like the energy that is derived from light, from photon absorption, will actually drive that hydrogen translocation from inside to outside. So again, there is all complicated electron transport chain that we will cover later. If we talk about utilization of hydrogen in cytoplasm, this is very specific for organisms that actually live in acidic environment. So as we already know, oxidation of various substrates, which can be organic and inorganic, will lead to extraction of electrons and their transport through electron transport chain. So all electron transport chain is followed by a series of oxidation and reduction. But what happens when electron transport chain is not ended with oxygen as terminal electron acceptor? So again, if terminal electron acceptor can be something else, can be nitrate, can be sulfate, can be carbon dioxide. In my previous class, in my next class, I talked about that. That's called anaerobic respiration. So if oxygen is terminal electron acceptor, then reduction of oxygen will create water. But if sulfate, for example, is, then it will be hydrogen sulfide then, or methane. So all of these oxidoreductions are followed by proton transport across membrane, and the gradient form. So, but what if organism is thriving in low pH? Those organisms are called acidophiles. So they have to protect themselves because they are living in area where outside there are many, many, many hydrogen ions. It's very acidic. So they don't want to open their gates to transport proton across the membrane because that's how they can actually let other protons come in and completely kill their organisms, which means they consume protons inside of the cytoplasm. They're not opening gates. So what that means. Inside of the cytoplasm, they usually have some organic acids, and those organic acids will pick up protons that are coming in from outside or from inside, but mostly from the outside, and that results in production of hydrogen and carbon dioxide, but they're not pumping protons across the membrane. That's why they have inverted membrane potential. In these organisms, actually, um, outside is more negative, inside is more positive, because they keep protons inside. That's why it's called cytoplasmic utilization, organic acid that picks up protons. Last but not least is the oxidation of substrate in periplasm. So very, very interesting mechanism. And it's tightly related to hydrogen, molecular hydrogen as a source of electrons, because hydrogen is highly reduced. And people who took Bio 322 learned already that the hydrogen was one of the first energy sources because it was highly abundant, and organisms developed hydrogenases to extract electrons from hydrogen. So, and this hydrogenase is actually living in periplasm. So that in periplasm is the place where hydrogen is oxidized, which means electrons are released. So again, because it's an electron transport chain, those electrons that are released from hydrogen by hydrogenase will be delivered to the further electron acceptor, but hydrogen that will be generated will not go anywhere, will just remain in periplasm. So one of the great citations that I heard in the C1 meeting a while ago was that life is nothing else but electrons searching a place to rest. And I know that that's not probably something that we ever think about when we mention the life, but this is how microbes learn, and that's how they see the energy formation. So finding a place, finding something to take electrons from, and finding something like that electron from rest to create energy. With that, again, any questions so far before we move to the membrane transport, which is really cool. It's actually amazing. The membrane transport is so easy. No way you won't get it. Yes, Ellie.
I don't know. I didn't hear about the difference between
Okay, so I will be very careful on this one. There is the difference is that, so direct proton translocation is part of it. So terminal oxidase that does that high direct hydrogen translocation, that is part of EPC. So that's why I want to say there is no difference. There is a difference because in complexes 1, 2, and 3, there is a series of oxidoreduction that are happening over time and that happen simultaneously. When we think about terminal oxidase, like cytochrome oxidase or terminal oxidase, that's one-time thing. Like electrons are released, proton is translocated, and that's the end, and the next one in line is ATP synthase, and that one does not accept electrons, like that there is no redox soup, because the complex one, which I'm not mentioning here, is ATP synthase. So we have complex one, which is NADH dehydrogenase. We have complex 2, which is lactatinide dehydrogenase and choline. You have complex 3, which is cytochrome oxidoreductase, and you have complex 4, which is cytochrome, which is terminal oxidase. Is also related to cytokines. So complexes 1, 2, and 3. You have all the electrons are released from these junctions. This gets reduced. And this releases electrons, gets oxidized, but then the other one gets reduced. Then the third one does the same thing. The musical complex 4. If you release it will get reduced. So the electron get reduced, and that's it. But it cannot. All that proton gradient will now be used in ATP synthase, which is complex 5. So there is no that redox group that has 1, 2, 3.
and at the end.
Kind of. Yes, I'm very careful to say it's at the end because it's the dynamic process. Because when you are finishing one round in complex 4, the next one is already running and it's coming. So it's in physiology, it's nothing can be considered as a black and white because it's a dynamic structure. So it's very hard. I know it's hard to imagine the 3D, but this is the rough explanation that helps. Yeah,
It's the only one that has the option. That's or the option that's by
exactly. Yes, that's a good point. Yeah, so it's the only that complex four is the only one that actually, like, literally, if you think about it, think about the iron and copper literally like holding that oxygen, like it's in two hands. It's holding it, and you can see later when we talk about the changes in respiration when oxygen is not present, there's thermal electron acceptor, there will be no that proton pumping because there will be nitrogen as well. And that's one of the modifications in E. coli, for example, when it goes on negative. Yes. Okay, so the question was how acidophilus maintain their starting acidity levels. Again, this will be very detailed covered when we talk about membrane stress, when we talk about bacterial stress in unit 5, but briefly, the way how they do this, they have something called donor potential, which means inverted membrane potential, which means instead of like normal organisms having way more H+ ions outside positive charge, and way more negative inside, these guys actually do opposite. So it's more negative outside and more positive inside. That means that they're literally holding H+ ions inside, and whatever they do, so if they open their gates, if they open their protein transportation, what they would do, they would extract more protein, they would send more proteins in the acidified environment. But instead, what they do, whichever proton comes from outside, gets inside, but they make organic compounds, like organic acids, that literally grab that hydrogen ion and prevent it to acidify cytoplasm. But if they open a gate and more ions come in, they will spend energy to make more organic acid, again, they will die. So they literally close the gate. And you will see later when we talk about different adaptations in acidophilic environment, because not all organisms are real acidophile. Some of them learn how to be acidophile. The way how they survive, they make something called proton channels, so proton pumps. So they literally do, let's say, push proton out of the cell. So if a lot of protons come in, that is not suitable for their organism, they can produce more organic acid, but in order to maintain that environment, they just create a pump that pushes proton outside. So that's how they survive. And that makes sense. Yeah. I know you guys, you all are getting confused now. It will be just doing. I knew that. But don't worry, my transport is easy peasy. There is nothing too high. That's easy to memorize. Simple, very simple. What's happening on membrane? Transport. Transporters. This is the beauty of physiology. There are many types of transporters, and microbes usually develop them as adaptations, and they learn how to live in different environments with different nutrient sources. And all those transporters that we will cover are used to get molecules in and out of the cells. So you will find a different groupation of transporters in different publications. So for me, it was really hard to decide how I want to group them for this class. Luckily, I imported those lectures from other profs, so I didn't really have to think a lot. But I had a hard time grouping them in my head when I was a student. So again, there are different groupations. My former boss was grouping them differently. Dr. Stein was grouping them like this. So first one is channels. Channels are transport. And most of the time we talk about integral membrane receptors. That's where the transport happens. So channels are the first and the most simple one. Secondary transporters are the second and a little more complicated one. Although they're not really complicated, there are two different types of examples that we will cover in secondary transporters. So the easiest one are ATP-linked transporters. As the name tells you, they need they use hydrolysis of ATP to drive something in and out of the cell. And last but not least is PTS, or phosphotransferase system, which you already heard about when we talked about catabolic repression and phosphorylation of glucose versus lactose, E2, ABC. So this will be fairly easy. So in terms of channels, so what is channel? Think about it as a protein that is embedded in membrane and it has like a little hole that drives ions to it. So that's a channel. That's literally how it is. And usually it's compounds that are moved through these channels have ions. So that's easy. And we'll talk about example mechanosensitive channel. Second one are again carriers, and we call them secondary transporters. Again, some people are calling them carriers one. What is characteristic for them is that they're using EMF-derived energy. So they need proton is involved in translocation of molecules via these transporters. So usually one of the molecule, ion, which can be transported across the membrane. So if one molecular ion is transported across the membrane, then this is uniport. Or if you are transporting two molecules, that can be symport if they are going into the same direction. Sometimes one molecule comes in, the other comes out, and that's antiport. And in very often, and in examples that we will have in class, one molecule is iron, hydrogen, the other one is sugar, and they are linked to each other. So the transporters that we will cover will be TRAP and lactose transporter. So again, either uniport, symport, or so those TRAP transporters that I'm talking about, they are ATP linked and hydrogen linked. So they're like combination, and we will use them as example. So they're secondary transporters, or somebody calls them carriers one. We will call them secondary transporters. Carriers two are also transporters, but they are unlike carriers one or secondary transporters. These guys are using hydrolysis of highly energetic phosphate bonds, which can be either ATP, which you will see most often, or phosphinophile, again, something that you saw when we talked about catabolic retraction and the lactose vessel glucose utilization. So again, ATP linked use ATP hydrolysis, but not PMF. That's a big difference. So this one, usually it has to be some ATP degrading and synthetic activity involved in this, like hydrolytic activity. But if it is phosphate based, that's still carrier, but it doesn't use ATP, it's using, again, we talk about that when we talk about metabolic repression. The example of, now we're moving back to the example. So channels, the easiest one. So the example is mechanosensitive or MS channel. So these channels, this is usually passive transport, and it transports small solids, usually ions that have different charges and sizes. So different ions has a different channel, and it's based on the size, like that's how big ion is. The example that we will have here is the mechanosensitive channel, which is usually developed in organisms in order to regulate their turgor pressure. So when bacterium is running into the hyposmotic environment, which is like freshwater, that means hyposmotic, that means there is no high amount of actively dissolved solids in it. That's why it's freshwater. Like hyperosmotic would be like something full of salts. So when bacteria runs into the hyposmotic environment, this will be hyposmotic shock that will activate MS channel. And then the gate on MS channel, that's how it looks. It has a teeny tiny gate. It looks like I don't know English words for that, but it reminds me on some like a clip that you use when you want to hang your laundry on the string outside. Yeah. And that's literally how it functions. We don't use that anymore because we have dryer, but I used that when I was a kid. So that's what it reminds me of. We call that clip. So the clip will then open when it's a osmotic shock. So now the clip will form the gate, and solutes will literally solutes that will now be inside. So there will be more solutes inside of cytoplasm than outside because it's hyposmotic environment. So those solutes will now from cytoplasm move to outside of the cell. So it's like a safety valve. literally, that protects cells from lysis and regulates its turgor pressure. So it's trying to make like equilibrium between inside and outside of the cell. So this is very easy. One of this mechanism is fairly easy for transport, and it's not hard to understand. The one of the confusions here is whenever we compare track transporter and lactose ferment. So they both function with hydrogen ions, which means they're both secondary transporters, which means they both use PMF. But the order of events is different, and I will tell you each of them. So track transporter is the first one that we will cover, and that one transports C4 sugar, like succinate, like fumarate, like something like that, something that has 4 carbon compounds. So it still has the general constitution, like transport of salts inside and outside of the cell. But what characterizes these transporters, both and trap and lactose, is that they exist in 3 states. Closed state, so when carrier is in closed state, so that's the first state. In that closed state, the domain of the carrier that is meant to bind the substance, like sugar or lactose, which is also sugar, it's located outside. So it's exposed to the outside. So carrier is in closed state, but gates are open and ready. Well, not gates, but like a binding domain is ready to accept the substance, but that's a closed state. Once when solute bind to the solute binding domain, that solute binding is causing something called conformational change, and literally the carrier is trapping that compound, and that changes its conformation. And this is now like that, literally change of state. It's not really closed anymore. It's in somewhere in between. It's transitional. But in the moment when carrier gets to be in the open state, that is the moment when now this solute, alongside with solute binding domain, is actually transferred to the inside of the cell. So it's exposed toward the cytoplasm. The gate is opening inside and the solute is released. So think about this receptor as something integral in membrane that is fulfilled with very flexible material. And when solute is binding, that changes conformation, and as it's sliding through the receptor, it's just doing this, and changing the conformation of the receptor. Then the outside coming inside and get released inside. Ready to accept the new one. Again, very dynamic structure. And this exists for all secondary transporters, like those, when these are 3 states where the medium one is intermediate and trans. And the end result is solute is derived to same side. So they're relied on proton motive force. That's why you will see this example of trap transporter, which is a sugar transporter, and you will see why it's relied on PMF. There is no ATP involved there. So periplasmic component is similar to ADP transporters, similar to ATP link transporters, but it's not. They just have similar structures. What is characteristic of this transport is that it functions as a symport where hydrogen ion is bound alongside of sugar, and because it's hydrogen ion, we say it's PMF link. So it needs to have that PMF grade. So how it functions, where the whole process starts. So initially, when it's ready to take C4 sugar, the transporter is in outward facing conformation. Like it's open, it's not state, but it's open to accept sugar. What's happening before binding? So there is this periplasmic binding protein on trap transporter. So it's like it literally looks like a little tail. It's part of the transporter that kind of screams through sugar in periplasm. Once when it detects sugar, sugar is the first one that binds, it brings it back to the membrane component. So it searches, screen, and brings it down where the transport will actually happen. Then, once when the sugar is bound, is brought up to the transporter, this is a signal for H plus ion to come and to facilitate movement of sugar through membrane. So first you have sugar, then you have H plus ion that facilitates it. As sugar is moving through the transporter, the transporter now goes through conformational change. We have that intermediate transport. And finally, sugar is moving downside as like through the transporter. And finally, the transporter is in so-called inward facing or conformation, and sugar gets released inside of the cell. So again, those are some C4 sugars, which are like succinate, fumarate, malate, some sugar-based acids, that kind of sugars. It's not for glucose. Glucose has its own transport. But it's a simple. So sugar first, then hydrogen ion, and you get acid. Another secondary transporter, which is PMF linked, is LAC-Y, symporter, and the name should already tell you that this is a symport, which means two molecules moving into the same direction. And you again have conformational change in response to solid binding. So LAC-Y is a transporter that's the same like trap transporter at the beginning in outward open conformation. But unlike trap transporter, LAC-Y now actually binds first hydrogen ion. And that now it's opposite. So in trap transporter, you first had sugar binding, like that part of transporter that moves through periplasm and finds to sugar, brings it back, and then hydrogen binding. In this case, in LAC-Y case, you will have first hydrogen, you will first have hydrogen and then lactose binding to the transporter. So just the order is different. Once when lactose is bound, because lactose is something that moves through, LAC-Y goes through conformational change, and you can see here in picture how it literally like changes the shape of it, and it's taking inward open conformation. So lactose is released inside of the cell, and hydrogen will leave transporter, so it's ready for the next round. So this was the example of two examples of symport, two different, so actually sugars, different types of sugars, but still symport and PMF involved. Drug antiport is something that is very common in bacteria that are resistant to antibiotic, and antibiotics are often toxic drugs for certain bacteria. So when bacteria runs into the toxic drug, it will try to remove it from cell, and that's where antiporters will do the job. So it's not only that, it can be also ATP linked, but there are some antiporters that will bind hydrogen at periplasmic side of the membrane, and next step is binding a drug from cytoplasmic side of the membrane because drug already came in. The common thing is that antiporter is going also through conformational change. Conformational change is caused by hydrogen and drug binding. So this is similar like lactose, also hydrogen binds first. And the next step, drug is released in periplasm and hydrogen will leave antiporter. So when hydrogen leaves antiporter, antiporter is ready for next round. So it binds hydrogen, then binds drug and so these were secondary transporters that were all linked to the PMF, proton multiports. There is hydrogen involved in all, regardless of whether they are importers or antiporters. Another type of transporters, which are ATP linked or PTS, like phosphotransferase system, they're linked to the high energy bond hydrolysis. So the first one that we're talking about briefly are ATP linked ABC transporters. You will see them very often as a multi-drug assets. What they have, and what is very common to see, is periplasmic binding protein that will initially screen for the substrate in periplasm. And once when it finds substrate, it will bring it to the membrane-bound compound. So you see the similarity between trap transporters. But what will drive the substrate into the cell will be ATP hydrolysis. So it must have associated ATP hydrolytic compound. So usually there are a few proteins that are spanning both membranes if we talk about gram-negative bacteria, which you will see later. In phosphotransferase or PTS system, it's not ATP hydrolysis that drives molecules. It's phosphoenol pyruvate hydrolysis, and we already saw that in catabolic repression, and we will see it again today. So, how the ABC system acts? So, in ABC transport mechanism, so when there is no drug, ABC transporter is in so-called invert or open state. When drug binds to the domain located in cytoplasmic side of the membrane, so think about the double register here, and now drug binds on that drug binding domain, that causes ATP molecules to bind to the specific component of transporter that has ability to do ATP hydrolysis. So ATP bind, and again, transporter is still in closed state. When drug is tightly interacting inside of the transporter, ATP is already bound. So you have to have drug bound and you have to have ATP bound. And you have to be tight. Without ATP, drug cannot be exported. So once when drug is there, tightly bound, then ATP hydrolytic component of the transporter will do hydrolysis of ATP to ADP and PI, and drug will be released to periplasmic side of the membrane. Along with the drug, because ATP is already hydrolyzed, PI component of ATP, because ATP is hydrolyzed to ADP and PI. PI will be released to periplasm, and now transporter will be actually ready to start another round. So both components have to be released, so transporter is empty and ready to take drug, and ATP perform ATP hydrolysis and use it to export the drug. The next in line is phosphotransferase system, which is PTS. Again, in this case, you probably remember of this component from what we talked when we talked about E1, E2, E2ABC, and HPR. So this transporter is glucose binding transporter. So when glucose binds, the full structure of PTS transporter will initiate the mechanism of glucose uptake, and glucose will bind to the surface of the membrane. In order to be metabolized, glucose has to be phosphorylated. That's where this phosphorylation cascade comes into the game. So when glucose is bound, the cascade starts from down, from inside the cell, where phosphoenol hydrolysis, PEP hydrolysis, provides phosphate group. So PEP now gets hydrolyzed, and the phosphate group gets attached to the EI or E1. I believe it's 1. So E1 portion of transporter, that's soluble, and it's inside of the cell. So once when that phosphate group is added to E1, E1 becomes phosphorylated E1P. E1P now has a phosphate group that will be delivered to next component in line, which is called HPR. Where HPR accepts phosphates and becomes phosphorylated, as the name says, is HPR phosphate. The cascade of phosphorylations and dephosphorylation is still ongoing. The very important step in this cascade is transfer of phosphate group. So HPR now becomes dephosphorylated, and phosphor group now goes to the E2 components. And there are 3 components of E2. We talked about A, B, and C. So this is where it will become very familiar, because we already briefly mentioned this. So first, phosphate will go to the A component, and A component becomes phosphorylated. And A component will give it to the B component, all soluble. Glucose is actually waiting to the insoluble A2C component that is in the membrane. And phosphor group goes from A, B to C, which is in the membrane, and that's where glucose waits to be phosphorylated. So when E2B gives when E2B gives when E2B gives the phosphate to the glucose, so it's not giving it to the C component, it's giving it to the glucose. E2C is just in the membrane where glucose is sitting. So glucose 6 phosphate is now ready to be metabolized, and that's when the glycolysis starts. And then you will have fructose 6 phosphate, fructose 1,6 bisphosphate, aldolase, and everything else that you will cover in Unit 6. So many of these detroit transporters are known as multi-drug efflux pumps, and many of clinical bacteria isolate actually are gram-negative resistant pump organisms that have multi-drug efflux pumps. And there are 6 known families in these efflux pumps. You will see them commonly in pathogenic isolates. ATP-linked pump is ABC transporter. MFS, it's a major facilitator superfamily. This one is not ATP linked, but the reason why I'm mentioning it is because it's common for drug resistance. So they're proton multi-force link, or you have mates, which are also PMF linked. So you do need to know which type of energy is carrying it. So you do need to know that ABC is ATP linked, but it's enough to know abbreviations. Mate is PMF linked. And the small drug resistance or SMR is PMF linked. R&D, I memorize this because I memorize this as a research and development department. So it's also PMF linked. So PACE is also PMF linked, which means it usually involves hydrogen ions. So notice that ABC transporters are the only one that actually use ATP energy. It's quite diverse among clinical isolates, and when you start searching for your papers to present, you will probably see, if you talk about antibiotic resistance, you will probably see multiple variants of these 6 families. This is just a general structure of these pumps. They have they look quite different. So the one that I really like are these 3-part pump that has 12 C in outer membrane. So this one is actually spanning the full membrane, and this 12 C is a very common structure that you will see in many gram-negative bacteria. And this outer membrane pump 12 C is very commonly linked to type 1 secretion system we will cover in the next unit. So they are, in my opinion, the most interesting. But the other one are just similar structure. They all use some sort of also usually linked to. Again, I really like this tall C outer membrane protein. So you can really even it can be part of ABC transporters. It can be also part of major facilitated superfamily, but it's the same tall C. So it just has different inner membrane structure. The last but not least is, and I think you will have a time for your lecture, is P-type ATPase, ATPase for potassium transport. So we talked about this KDO pump when we mentioned this KDO pathway, but it's not the same type of transport. This one is specifically involved in potassium transport, and it requires phosphorylated intermediates. So it has 2 states, phosphorylated and dephosphorylated states, E1 and E2. And depending on phosphorylation level is dependent on the presence of potassium in microbial cell. So the substrate is potassium, and depending on the level of potassium, phosphorylated medium will respond. Phosphorylated intermediate will respond differently. So the way how cells regulate the level of phosphorylated versus dephosphorylated transporter is through proteases in cell. And the name of the receptor is called KDO KDP, sorry, KDP potassium transporter. So don't get confused with KDO. It's a different type, but students often confuse it because it's similar, like KDO versus KDP. And KDP potassium transporter is very universal in bacteria, and it actually has three subunits. Well, three membrane spanning subunits, but they are like 6 proteins. So each of subunits is a dimer that it would remember. That's why we say A2B2C2. And the reason why I'm mentioning all these three subunits is because each of them is related to the specific function. For example, B subunit is coupled with phosphorylation. So B subunit is where phosphorylation actually happens, which determines A1 versus A2, E1 versus E2C. And because it's related to phosphorylation, that means that the energy for transport is related to KDPB subunit. So what's the mechanism? So again, I told you that there is E1 versus E2 state. So E1 is a non-phosphorylated state. Logically, E2 is phosphorylated state. So if potassium is not present, you will see E1. That will be dephosphorylated. When potassium comes in, there will be certain conformational changes from E1 to E2, and phosphorylation will happen at KDPB subunit. So that B subunit will accept phosphorus. Now, when transporter comes from E1 to E2, the activity of transporter will change, which means it will be able to bind potassium that needs to be incomplete. So potassium will be able to bind only if unit is phosphorylated on KDPB, and potassium loads on transporter. And what the next step will be, like you have to now, when it binds potassium in phosphorylated states, in order to release that potassium in cell, you have to dephosphorylate B subunit. So there is phosphatase that removes phosphor from E2, and the transporter is now from E2 into E1 state, and potassium is released into the cell. And the energy for transport, of course, originates from ATP. The transporter is present in inner membrane, it's integral membrane transporter, and it mobilizes potassium from periplasm to cytoplasm. So this transporter is related to maintaining the potassium level inside it. Yeah? And the way how it's maintained, that means that what that means is that those transporters must have some sense of how they will detect whether they need to be phosphorylated or dephosphorylated. So they will change state from E1 to E2 depending on the level of potassium. So they have to have some sensor. So the sensor that senses the level of potassium is nothing else but sensory kinase. And sensory kinase, again in the membrane, is phosphorylating itself, which means autophosphorylation, and it does that depending on the levels of potassium in cell. So it's called very creatively KDP sensory kinase, and what this kinase is sensing is the difference in salt, the difference in potassium level, like osmotic sense. It responds especially to potassium. So when there is not enough potassium and cell needs to import something, so the lack of intracellular potassium, that will trigger so-called two-component system, which means phosphorylations of different levels. And this sensory kinase will complete something called autophosphorylation. So how is that? So again, sensory kinase feels it, the low potassium. Now that means we have to do phosphorylation at KD. This should be KDP. Sorry, my mistake. It should be KDPB. You see, I also made a mistake. So sensory kinase is doing phosphorylation at KDPB. So that's how it's feeling. So sensory kinase is actually KDPB. And this is how it looks. KDPB IMMAC, integral membrane receptor. So now when KDPB gets autophosphorylated, now it's KDPB, brings low calcium, the phosphate group from the sensory kinase is transferred to the so-called response regulator, which is KDPE. So what phosphorylated KDPE does is bind downstream to the inside the cell, bind to the promoters that will activate transcription of genes to synthesize more and more potassium transporter. And that those transporters will now get embedded in membrane and they will get open to import potassium instead. When potassium reaches high level, proteases will degrade transporters. Very similar to potassium, again, you have KDP, P and E, is iron link transporter. So notice that you still have that spanning of inner and outer membrane. So let's say that siderophores, which are example that I'm using. So what they do, they are involved in iron transport. So they chelate iron 3+, as you can see. So pack A is in integral membrane, in outer membrane that defines iron. So pack A is actually now sensor. So it's analog to KDPB. So it senses iron, and what you have here, you have this tone B. Again, this is not the same like tone C. So this tone B is just a linker. Tone B is linking these XBDE proteins that are using PMF to bring iron into the periplasm. So there is a teeny tiny PMF involved here, but it's also related to ATP hydrolysis, because ATP hydrolysis is happening inside of the cytoplasm, and it enables actually this transfer from periplasm, actually from outer membrane to periplasm and then to cytoplasm. And all of these transports that I just explained to you are involving some sort of sensor. And in first one, you saw sensory kinase that gets autophosphorylated. And if you remember, there was one set of phosphorylation on sensory kinase, and then that phosphate group was transferred later on to response regulators. So this is something that we call two-component transport, and this is controlling mechanism at transcription level. So the structure that usually senses the drug, which was KDPB in our case, it's sensory histidine kinase. What means histidine kinase? That means that that initial autophosphorylation is happening on a histidine that is present in transporter. So the first step, and this order really matters. I really want you to understand this. The first step in this process is the drug binding to periplasmic domain of sensory kinase. So drug needs to bind to activate that sensory kinase. When drug binds, that activates sensory kinase, which is the periplasmic site, that will lead to the activation of cytoplasmic domain of sensory kinase. And what you will see is autophosphorylation. So again, sensory kinase and cytoplasmic domain will phosphorylate itself. This histidine is usually on cytoplasmic side. That phosphorylated kinase, like sensory kinase and histidine, is now able to complete phosphorylation of something called response regulator, which is inside of cytoplasm. And that response regulator is usually phosphorylated or aspartate, because only aspartate phosphorylated response regulator can go down and act on certain genes to initiate certain transcription. That's why it's called two-component system. You have sensory kinase that completes phosphorylation of histidine, and only in that state it's able to transfer phosphor group to the aspartate of response regulator, which later on will cause transcription of certain genes. And that would be all for this class, and we don't have time for Marissa's lecture, and I'm very sorry. I knew this will happen. I'm very sorry.
Okay, the examples, if you want to get ahead of it, are already posted, so you can read through it. But also you have plenty of time. There's more than a month before assignment